Thank you very much for the introduction. So, first of all, I want to thank the organizers for this opportunity. And second of all, I would like to advertise IBSBC because it hosts one of the two French Instruct centers. And if you have a difficult protein to express and you want to try insect on mammalian cells, or if you want to characterize a sample using a battery of biophysical instruments, or if you want to collect high resolution data, you should really come. We have a really awesome environment. So as Arnaud pointed out, I'm interested in transcription and translation, and conceptually those are two distinct steps, right? They are the minimal two steps that need to occur in gene expression, and in a biochemistry textbook there are typically two separate chapters, and in molecular biology there are also two separate fields. But in prokaryotes, the two processes are actually tightly coupled. And so this is another famous Miller spread, we saw one earlier from Arnaud, which depicts a bacterial lysate visualized by an electron microscope, and the horizontal line here has been interpreted as being a protein-coding gene that is actively transcribed by multiple polymerases. One is highlighted with an arrow here. And as the transcript increases in length as we go from left to right, more and more of these black blobs join the transcript, and those, of course, have been interpreted as being ribosomes that trail the transcribing RNA polymerase. Now, a few years ago, my lab was able to contribute a slightly higher resolution image of this, and we characterized RNA polymerase shown in red here, coupled to a translating ribosome shown in yellow and blue for the small and large subunit respectively. And what I really want to highlight here is by organizing the transcription and translation machinery in this supramolecular assembly, you see new functions that emerge, which are very difficult to predict when you study the machines in isolation. So for example, if the RNA polymerase is coupled with a transcription factor called NUSG to the ribosome, it gets aligned to the ribosome in such a way that the mRNA gets directly fed into the ribosomal helicase, and this ensures that the mRNA stays single-stranded. And you may remember from your undergraduate education that in bacteria, RNA secondary structures like hairpins can terminate transcription, but as long as you have a ribosome traveling behind you, that's not what you want, right? Likewise, one of these domains of NUSG, which binds to the ribosome, also recruits transcription termination factor Rho. And so for the same reason, as long as you mask this domain by a co-translating ribosome, Rho cannot be recruited, and so transcription proceeds. But if the two machineries get closer to each other, they can collide. And it turns out this also has important functions. So for example, a colliding ribosome can release RNA polymerase from regulatory pulses. And more recently, it was shown that the translating ribosome can also remove RNA polymerases that are stalled on DNA lesions. So what I really would like to drive home here is that by organizing these two machineries, there are all these new functions that emerge that are very difficult to know beforehand. But today I'm going to focus on the very first step, when the ribosome gets recruited to the polymerase. And so again, biochemistry from Irina Artimovich's lab and Niels Waltz's lab already pointed more than 10 years ago to the fact that the polymerase is able to stimulate ribosome recruitment to the base of the mRNA. So we wanted to understand how that can possibly work. So not everybody in the audience is familiar with ribosome architecture. So let me introduce a few things that I would ask you to keep in mind. So most of the time I will show the small subunit in this view. It's basically from the perspective of the 50th subunit. The top part is the head, the bottom part is the body, and it's separated by the mRNA binding channel, which accommodates the mRNA shown in magenta. The 3' portion of the mRNA binds near the entry tunnel of the ribosome. The 5' portion of the mRNA leaves the ribosome near the exit tunnel. And in between, you have the 3 tRNA binding sites. Now, in the exit tunnel, you also find anti-Shang organo sequence. And you remember from your undergrad, 16S rRNA encodes the anti-Shang organo. It base pairs with the Shang organo in the mRNA, and this localizes the start codon somewhere near the P site, so that the initiator tRNA can bind the start codon and establish the correct reading frame, which of course is absolutely crucial. In addition, there is a ribosomal protein called BS1, also near the exit tunnel. And this one has a number of RNA binding domains and has been shown to be able and recruit mRNA unfolded, and it's likely responsible for efficient translation of most, if not all, mRNAs in bacteria. All right. So in a translating expressome, the polymerase shown in red here would be localized near the mRNA entry tunnel, because of course it extends the 3' end and feeds it into the ribosome. So one thing that I would also like to emphasize is that since the late 60s, we know that the 30S subunit can interconvert between an active and an inactive conformation. And Harry Noel's lab has shown in the mid-80s that this, that inactive versus active confirmation actually is characterized by a misfolded decoding center, which is responsible for monitoring tRNA and mRNA. But then more recently, Kevin Weetz's lab has shown, using in-cell shape, that maybe most, if not all, 330S subunits adopt this inactive state inside the cell, and so they need to constantly be activated. And also, it's important to realize that this inactive conformation is also part of ribosome assembly. So a late maturation state mimics this inactive state of the 30th subunit. And last but not least, Joey Davis's lab at MIT has shown last year, using extensive classification of E. coli lysates, that a significant fraction of the 30th subunit adopt this inactive state, consistent with the earlier chemical coding data. So when we started out to study how the ribosome gets recruited to the nasal mRNA, we first thought we're going to use a simple system. We started with inactive 30S subunit, and we did not add initiation factors, and the rationale was that we wanted to capture the early state in the recruitment process. And so when we did EM on this, we basically found three populations. An inactive 30S subunit, and I will explain to you in a second how we know that. An inactive 30S subunit bound to RNA polymerase near the entry tunnel. So in a way, this is what we expected. A very small population of small ribosomal subunits with a tRNA bound to the start codon. The mRNA has been properly accommodated in this case, and the ribosome is fully active. And then a relatively large fraction of particles that we did not expect, but the ribosome was active. But RNA polymerase, rather than being localized near the entry tunnel, it was localized near the exit tunnel of the ribosome. And I will now tell you about unpublished work where we basically confirmed all these states, but I think it makes more sense if I put them into a larger picture. So, what we did next was, we wanted to understand what happens when you add translation initiation factors, and can they possibly play a role in ribosome activation. So in this case, we formed an RNA polymerase elongation complex with a nascent mRNA that encodes a strong nano sequence. We added initiator tRNA and the three initiation factors. These are here, they bind to the A site. IF2 recognizes the amino acyl end of the initiator tRNA, and IF3 prevents association of the 50S subunit and also selectively destabilizes non-initiator tRNAs. And so when we did cryo-EM of this complex, we found at least 11 intermediates. Okay, and I will try to walk you through them now. We ordered them based on whether the 30S subunit was active or inactive, based on the number of initiation factors or tRNA that were present, and based on the position of the mRNA. And so I think what I would like to highlight here is this molecular choreography of this assembly process of the ribosome on the nascent mRNA. And I think for the first time we can also watch how the mRNA gets accommodated on the ribosome in a stepwise fashion. Okay, so let's start at the beginning. As a control, we did EM on inactive 30S subunits without any initiation factors. And basically what we found is that 100% of the ribosomes are inactive, and they adopt two different inactive states. In inactive state 1, this prominent ribosomal RNA helix called helix 44 is localized in the exit tunnel of the small subunit, and it seems to interact with ribosomal protein S1, which is this RNA binding protein. In inactive state 2, the helix 44 has relocalized onto the subunit interface site, so this is where you find its proper location, but the top part of the helix has not yet folded, and the top part actually makes the decoding center. So the decoding center is not yet recognized. And so I just want to emphasize, using this more, the quite dramatic conformational change that has to occur as helix 44 relocalizes to the subunit interface site. So now the situation changes when you add initiation factors, because instead of having 100% inactive ribosomes, we're down to something like 4%. We still find inactive state 1 and 2. They look essentially identical, except that one initiation factor, IF3, begins to bind to the ribosome. The N-terminal domain binds near the shoulder, but we don't see the C-terminal domain of the initiation factor. So this tells us that IF3 might be the first one that binds to inactive ribosomes, but on its own, it might not be able to activate them. The situation changes when IF1 enters the picture, shown in blue here. It binds to the A site as expected. Now the C-terminal domain of IF3 also becomes visible. So both factors are visible in their full And Helix-44 is now fully folded, so the decoding center is ready to go. And so we think that these two initiation factors together can activate the ribosome, and it may be interesting to note that the IF1 and the C-terminal domain of IF3 bind in the same location as a ribosome maturation factor called RSGA, and IF1 also shares structural homology with RSGA. So it's possible that the initiation factors sort of play a similar role as these late ribosome maturation factors. So now the polymerase enters the picture, and what I'm showing you here is a low-pass filtered map. So I hope you can still recognize the head domain of the ribosome and the body, but in addition we had this sort of cloud of density that was completely featureless. But when you do focused reconstruction of this cloud, you can show that it's a transcribing RNA polymerase bound to DNA with an RNA transcript emerging from it. In a subset of particles, we can also see ribosomal protein S1. Remember, this is the protein that recruits and unfolds mRNA. In a fully extended conformation, it's this sort of long arm that can float around the shoulder of the 30th subunit. And the polymerase is sort of occupying this cloud, but what I want to emphasize is that it's not found in any distinct orientation with respect to the polymerase. Rather than that, it's presumably floating around. And now I want to zoom in onto the exit tunnel where mRNA interaction with the ribosomal RNA takes place. So what you see here is the mRNA in pink, the Shine-Logano sequence, annealed to the anti-Shine-Logano sequence shown in green. And what I hope you can appreciate is that this structural model, which shows what it looks like when the mRNA is fully accommodated, does not match the EM reconstruction. Instead, what you have to do is you have to rotate the Shine-Logano and anti-Shine-Logano sequence. And I will ask you to keep in mind to pay attention to what happens to the RNA 3' end. So as we rotate the duplex between Shine-Logano and anti-Shine-Logano sequence, the 3' end of the RNA, rather than pointing towards the entry tunnel, it now points towards the exit tunnel. And that, of course, is where RNA polymerase is located and where it sort of extends the 3' end. So if we look at a low-pass filtered map, we can also sort of trace the path of the RNA. It interacts with the RNA binding domains of S1. And if we zoom further out, you can see that the RNA presumably meanders around the RNA binding domains. And if we superpose the low-pass filtered map, you can see that this cloud of RNA polymerase surrounds S1. So our interpretation is that the RNA polymerase can interact with S1 at various positions, feeds the mRNA to the RNA binding domains of S1, and then S1 guides the RNA towards the anti-Shine-Logano sequence in the ribosomal RNA to establish the first contact. So we did some biochemistry also, and we can show, I would ask you to just focus on the last thing here, that if you add S1 to an RNA polymerase transcription elongation complex, only if you have a long enough RNA to provide binding sites for S1, you can form a stable complex between the two. Okay? So it seems that it's the RNA that drives complex formation between the polymerase and the ribosome. However, if you put them on a sizing column, S1 co-purifies with RNA polymerase core in absence of nucleic acids, but only trace amounts. We can only detect it by Weixlbaumer. So again, it's the RNA that drives stable complex formation, but nevertheless, S1 can presumably weakly interact with the polymerase and sort of help localize it at this nearly exit tunnel. Okay, so now IF2 enters the picture. So IF2 is the translation factor that recognizes the amino acylated tRNA, but here it binds actually without the tRNA being present. And it binds in a conformation that has not been seen before, which we call the central conformation. The mRNA is still in the delivery state, so the Shine-Logano anti-Shine-Logano duplex is still inverted, meaning the polymerase is still localized here. It feeds the mRNA to the exit tunnel. But interestingly enough, IF2 has a disordered N-terminal domain, and two alpha helices form that occupy the empty mRNA binding channel. And so we think that IF2 at this point senses that the mRNA channel is still not occupied, and it adopts this sensor conformation which blocks 50S joining. Okay, so in a way it makes sense. IF2 ensures there is no mRNA yet accommodated in the binding channel, so therefore the 50S subunit should not yet join. Now that tRNA joins the picture, it can bind to the P site. The mRNA, however, is still in the delivery orientation, so it has still not started to accommodate. So we think that the tRNA just comes and goes and samples the ribosomal P site for the presence of the mRNA. But if you zoom in, you can see the anticodon loop of the tRNA. Here is the anticodon. But this helix of IF2 is also still present because there is no mRNA yet there. Now the picture changes once the mRNA begins to accommodate. And so here we have two states that we decided to put in this equilibrium here, and that's because the mRNA adopts the same conformation in both of them. But IF2 now begins to accommodate another conformation that we call the extended one, which is compatible with 50S joint. So if we zoom in on the mRNA, we can now see that it started to base pair with the tRNA, so the AUG start codon is now properly positioned in the P site. But the downstream portion of the mRNA, so I would like to draw your attention to the comparison between the orange mRNA path, which is a fully accommodated mRNA, and the mRNA that we see in this intermediate, and so the density was really unambiguous. So the mRNA is starting to accommodate near the ribosomal P site because it pairs with the tRNA, but downstream of the start codon, it's still outside of the channel, so it has not been lowered down. And that's because IF3 in green and IF1 in blue are holding on to the mRNA before it gets lowered into the channel. I just want to highlight the conformational change that IF2 has to undergo between the sensor and this extended conformation that enables 50S joining. In both conformations, the C-terminal domain can hold on to the tRNA, but the G domain actually relocalizes on the surface to enable 50S joining eventually. So I've been a bit quiet about the polymerase, and that's because in all these states here, we don't really know where the polymerase is. It's presumably attached to the 3' end of the RNA, but because the RNA is quite flexible on its 3' end, we think it sort of hovers above the small ribosomal subunit. But the situation changes when the mRNA begins to accommodate further. Because now we can see RNA polymerase localized near the entry tunnel via transcription factor G that I mentioned earlier. IF2 can still oscillate between the sensor and extended conformation. So in this case, there are still helices that are inserted into the mRNA channel, but in the extended conformation, it's now ready to accept the 50th subunit. And so the RNA, the mRNA, has now further accommodated. So the P site codon is still bound, the start codon is still bound to the P site initiated tRNA. The downstream portion of the mRNA has lowered into the channel. That's because the polymerase is already in the right position. But the part around the A site codon is still held by IF1 and IF2. So this part has not yet lowered into the channel. So this requires release of IF1 and IF2, and that happens when the 50th subunit joins. And so we were quite lucky because our 30S preparation contained trace amounts of 50S subunits. So in the end, actually a small subset of particles formed at a 70S ribosome, where IF1 and IF2 have now left the complex. IF2 is still present in its extended conformation. The reason it's still present is because there is a GTP analog that cannot be hydrolyzed. So IF2 is basically blocked before GTP hydrolysis and is stuck on the ribosome. The polymerase is at the entry tunnel, it's feeding in the mRNA, and so now we've basically reached the end of the cycle, and the complex is now ready to enter the elongation phase of translation. And so there's two more things that I would like to highlight. So one is we teamed up with Niels Waltensnyder at the University of Michigan, and we basically asked, in our ideal system, because we use a highly idealized mRNA that has a very strong shiny organo and almost no secondary structure, can we still see an effect of the polymerase recruiting the ribosome? So Niels' lab immobilized either an RNA polymerase elongation complex with our ideal mRNA and labeled with a fluorophore, or they immobilized the RNA with complementary DNA, so that is without RNA polymerase. And then the 30S subunit also carried a fluorophore, and then they asked what is the weight of 30S recruitment to a transcription elongation complex versus an mRNA. And the effect is not huge, but again, keep in mind that this is a highly ideal mRNA. So even here, in presence of ribosomal protein S1 and in presence of RNA polymerase, you have the fastest rate of 30S recruitment to the mRNA. Okay? All of this was in vitro work. So the question is, does any of this occur inside the cell? And so for this, we teamed up with Julian Absilber in Berlin, And Yuri's lab, masters this in-cell cross-linking followed by mass spectrometry to characterize protein proximity. Okay? And so they use this approach to ask what proteins are near RNA polymerase in E. coli. And so they found cross-links that are consistent with RNA polymerase in this expressome position where it feeds the mRNA to the entry tunnel. But most importantly for this talk, they also found cross-links that are incompatible with this position, but are compatible with the position of RNA polymerase in the exit tunnel. And so that gives us some confidence that even in the cell, the polymerase interacts first through S1 to feed the mRNA to the ribosome so that initial Shino-Gana interaction can occur before the mRNA gets accommodated in a stepwise manner in translation and initiate transfer. And so with this, I would like to thank the people involved in this work. Most of the parts that I told you are still unpublished and were done by Uma in my lab, who's an extremely talented graduate student and is looking for postdocs now. So if you get an application from her, I would